cellular respiration cycles. So in the beginning we have a glucose, and we add energy in the form of ATP to this glucose, and it then breaks apart into 2-pyruvic acid, giving us 4 ATP and 2 NADH. The pyruvic acid then will either go to Krebs or fermentation, and the ATP and the NADH will then either be used in fermentation or ETC, depending on what the pyruvic acid has gone to. From here, we take a pyruvic acid and we break off a carbon, and that's released in the form of carbon dioxide, and we get NADH from that. That releases NADH, breaking it into acetic acid. It then combines with the enzyme CoA and to combine with another 4-carbon molecule, and that creates citric acid, and that citric acid doesn't need the CoA anymore, so you get rid of that. And then with that citric acid, we break off another carbon dioxide, and then we get which releases more carbon dioxide and NADH, and then we get a 4-carbon molecule, 4-carbon molecule. I'm sorry. And we break off another one, and we get a 4-carbon molecule, releasing more CO2 and NADH. And then with that 4-carbon molecule, we switch around its structure, and that releases, and we capture that energy release in ATP. It rearranges again to create FADH2, and then it rearranges once more and becomes NADH. Onto the electron transport chain. We have NADH power these facilitated diffusers, and that pumps 6 hydrogen through the inner membrane to create a high concentration above the electron transport chain. The hydrogens will then go through the ATP synthase, and that creates 3 ATP. And there's water kept on the low concentration side to help keep a low concentration, because when the hydrogens come back through, it creates a bond with the oxygen, and they make water, and we can use that water in our bodies. And it's exactly the same process for FADH, except for only 4 hydrogens are used and only 2 ATP are produced.